knows that you have to almost have to sit at their bedside 24 hours a day to make sure that simple things like, knowing which patient it is and knowing what drugs they're already on and knowing what they're allergic to is known by every caregiver that walks in that room. So we really face the more complex the condition and the more people involved in their care, the more chance there is for that care to be screwed up. So really the story of delivering the highest quality medical or dental care these days is not just about figuring out which implants work or which drugs work, but trying to get 10 different people who might touch that patient to all have the same information and be working in a coordinated way, and that is incredibly hard. I don't know if that addressed your question, but part of the answer. Yeah.
Controversial subject of 3 or 4 years ago, the orthopedist saying that you have to have lifetime antibiotics right away that had and all of a sudden just turned around and said you don't. Patients are confused.
Yeah. Yeah. So the question was about a dictate. Start up again. I let everybody have too good a time. We took too long, but that'll be okay. So I hope your batteries are a little recharged. We're going to have to discharge them again a little bit here. And we're going to talk about numbers, but we're going to talk about numbers in I think, a very different way than maybe you've seen before. Some of you have seen parts of this already. I'm going to talk about statistics, not so much from, here's a p-value, here's a this, here's a that, this is how you interpret it, even though we'll have a little of that, but give you some insight in the philosophy behind these statistics and why they exist, why they're misinterpreted, why they're misused, but really understand what they were intended to do and what they weren't intended to do. And this sort of level of analysis is something that interests people a lot sometimes. Sometimes they find it completely irrelevant to their day-to-day lives, but I enjoy it, and hopefully it will give you more insight into why sometimes things seem to make sense and other times where you're totally confused. And the answer is going to be, if you're totally confused, there's sort of a good reason. It's not your fault. One thing I want to say, just in follow-up to my previous talk, and someone came up, I'm sorry I don't know your name, and said, which is very common for all practitioners in any field, which is, this is great, but I have neither the time nor expertise to do any of these things you're talking about. What do you recommend for a busy clinician, or even a not so busy clinician who simply doesn't have the skills or the time to dive into this literature and sort it through? So I don't live in the field of evidence-based dentistry, so I don't have all the final answers to that, but I would say a few things. First of all, for those of you who don't know, how many of you know what the Cochrane collaboration is? Have you ever heard of that? A few? How many of you have not heard of that? Okay, the vast majority. So the Cochrane collaboration is actually almost not describable because it's so unusual. It's an international, completely volunteer collaborative of investigators around the world, and any one of you can be a part of it tomorrow. Just raise your hand, say, okay, now I'm part of the Cochrane collaboration. That has evolved over years, so it has procedures, but basically what it does is it does systematic reviews of interventions in all of healthcare. And it's organized around completely spontaneously forming review groups in particular areas, and sometimes they're very broad areas and sometimes they're very narrow areas. There is an area of oral health. And I'm not completely up to date on the Cochrane collaboration. That was the field in the past. It may have split into many subfields, but this may be the largest organized group of scientists in dentistry, in oral health specifically, that are devoted to looking critically at the literature and putting it together in the form of these things called systematic reviews. And what you will find when you go on their website is lists of probably tens or hundreds of reviews of interventions that you probably use, with very, very structured, similar formats for how they look at the literature. And it's really a sort of model for how to debunk or how to know what are reliably effective interventions. So I would recommend that you just go look on Cochrane Collaboration, COCHRANE, and see what you can find. Now, it used to be completely free and open. I'm not sure exactly what its status is now. I think it is still free and open, but it's worth looking at. So the second half of the answer is look for systematic reviews. The way to assess the literature, such as I showed you at the beginning, is to take advantage of the work that other people have done to look at a whole body of literature in a particular area and try to figure out whether it's good or bad or combinable or not combinable, and give you some insight as to whether there's consistency here and the quality of the methodology. There are people doing this work for you, and to the extent that you can, try to find those reviews. But it's the systematic part of it that's critical. Just the model of the expert sort of intoning the, his opinion or her opinion based on the articles that they have either selected or quote in a completely unsystematic way is not the way to go. And is those in medicine have shown, in a sense, long been abandoned, and because they're shown to be so unreliable, which is why the systematic part is so critical. They're really experiments, observational studies based on the published literature. Now, systematic reviews do not solve the problem of biased publication, selective reporting. There are all sorts of major problems with our literature that it's only people who do these actually ferret those out. But still look for them. They're better than looking at an individual study, because you'll be totally at sea. And any one study also depends on all the other studies that have been done. So I would encourage you to look for those sites. And also, I don't know how active your various professional associations are. Gary certainly knows, and other people in the room, about producing guidelines based on high quality evidence, or as high quality evidence they can find. Those are sometimes good sources as well, but those guidelines are only as good as the supporting literature, and if the supporting literature is very, very poor, the guidelines are not that good. And in most medical specialties, what they've now come to do is label their recommendations with different, they issue guidelines, but they rate them by level of confidence based on the underlying literature, A, B, or C. A being the highest level, B being some sort of intermediate level, usually based on some mixture of maybe of trials, maybe of observational studies. C really, in a sense, based on expert opinion, because the evidence just isn't there. So the more and more professional societies are doing this and being honest about where it comes from. So whether it's a high quality evidence-based guideline or just really the best we can do given an absence of information today. So I don't know the texture of your field as well, and maybe Gary can comment on this. Do you want to say anything right now? You'll speak later.
Yeah, I have a section.
Okay, great. So I think knowing your field, someone who knows your field inside out and knows what reliable evidence is better to talk on this. So let's start talking about statistics, and I'm going to start with this little research quiz. It says a well-done study is reported on a new electrical stimulator for endodontic pain control, and the authors state that it's turned out somewhat surprisingly. That is, they thought this wouldn't have more than a 30% chance of working out to be effective in actually reducing migraine pain. A completely incidental finding. The improvement was 15% with a confidence interval of 0 to 30% with a p-value of 0.05. So the question is, what is the probability that this association is real? Okay, so they used the electrical stimulator for dental purposes. They found out miraculously that folks who had migraine headaches reported a decrease in frequency with this stimulator, and it's reported with a p of 0.05. So The probability that this association is real, so I've given you three choices here: less than 75%, 75 to 94.9999, and 95 or greater. Okay, you get 4.5 seconds to think about it. Okay, so how many would vote for, now Gary can't vote, because, so actually the whole front table there can't vote because they've taken this test before. I won't tell you whether they got it right or wrong, but they've seen the answer. So is it less than 75%? How many? Fair number, okay. Intermediate level, 75 to just under 95. No one. 95. Okay, well, you're either shy or much smarter than most audiences that I lecture to, or maybe you saw it flash by before but the answer indeed is less than 75%. So why? Well, that will be the subject of the talk. But if you found it confusing or answered right but didn't know exactly why, you're not alone. Why? Well, because two things. Well, actually, let's skip here. Because doctors in general don't understand statistics, and this has been shown over and over and over again. So one of the first documentations of this was a study in 1981. Almost 700 pediatric residents were asked two questions. Just to ask two questions, the number one choice for the strongest correlation between two variables was 1.25. Now, if this was a quantitatively oriented audience, you'd guffaw. The highest value a correlation coefficient can have is 1. So answering 1.25, it's like asking what's the highest probability, and you would never say, what's the highest chance this horse can win? You wouldn't say 1.25. So that's not just wrong, it's horribly, egregiously wrong. The question P of less than 0.05 means that a result is also significant at, number one answer was 50%, and actually that wasn't necessarily wrong. But 25% said you can't make any such inference. Going forward in time, here's a study in 1993. 31 physicians were asked what the positive predictive value was for a test with this sensitivity and specificity with different prevalences. Now, some of you may not know all this language, so I'll just cut to the chase. The correct answer in those cases of low prevalence was 0.003%, 0.3%, and 25%. The answers the doctors gave were 50%, 55%, and 59%, showing that it would be very hard for them to use any information on diagnostic tests in a sensible way. In 1998, 300 practicing doctors were interviewed about methods they used to assess diagnostic test results. The vast majority used just sensitivity and specificity. And of those who used, which is really the most primitive way of using them, Bayesian methods, likelihood ratios, and ROC curves were used by a tiny, tiny fraction. And of those who used sensitivity and specificity, 95% of them used them improperly. Now, this is a real sign of the apocalypse here. This is a study that was published in 2007. You would think this would document progress. Here's what happened. Medical residents were given a test. 88% expressed fair to complete confidence in interpreting p-values, yet only 62% of them could answer a basic question correctly. But that's not the sign of the apocalypse. The sign of the apocalypse was that there was no correct answer that was actually offered. And the authors were unaware of this. What they called the correct answer was not correct. So there was not a single correct definition of the p-value offered on a test that was then published in the Journal of the American Medical Association. So that's really bad. That's really bad. And it shows you how difficult the p-value is to interpret. Actually, I grieved particularly about this because when I looked at this test, it looked astonishingly similar to questions I had seen before. The reason was they were derived from a test that we gave at Johns Hopkins when I was there for 25 years as part of a short course on statistics, and somehow this author got the test and gave it to their residents and then published the results. But they changed it in a few ways, and they changed the result on the p-value of the question to something that they thought was more right. And that I was the one who taught that part of the course. And they changed it to the wrong answer. So anyway, this you can't see. We've tried to sharpen up the screen just a little bit, but now it's smaller. But I'll just read down the list. The percentage of, these are 277 Yale residents, the percentage who got various things right. And there was hardly a single question that they got that more than 50% got right, except recognizing the purpose of double-blind studies. But even identifying a continuous variable, identifying an ordinal variable, recognize the definition of bias, interpret a standard deviation, that was just 50%. Recognize power, sample size, and significance level relationships, only 30%. Go up and down the list, almost everything is less than 50%. So you're not alone if you don't understand all this stuff, and it reflects really how health professionals are trained. This is not really part of the training. This is what R.A. Fisher, who's really one of the intellectual giants of the 20th century, having given us not only almost all the statistics that we use today, but also was the premier early geneticist. He developed the theory for blood groups and almost all the statistical genetics that we use now. And this is a speech he gave in 1958. He says, "I'm quite sure it's only personal contact with the natural sciences that's capable to keep straight the thought of mathematically minded people. I think it's worse in this country, the USA, than in most. I might be wrong. Certainly there's grave confusion of thought. We're quite in danger of sending highly trained and intelligent young men and women out into the world with tables of erroneous numbers under their arms, with a dense fog in the place where their brains ought to be. In this century, of course, they'll be working on guided missiles and advising the medical profession on control of this limit to the extent to which they can impede every sort of national effort." So he saw it coming, and I actually think it has gotten much, much better, but it still has a long, long way to go. So how did we get here? So this is really a bit of historical background, which I find fun. And just puts the sort of things we're talking about today in a very broad historical context. So the first person who wrote a book on evidence-based medicine was this guy named Girolamo Cardano, who actually invented the theory of games. It's 1525. He wrote a book on games of chance, but his real goal was to be the doctor to the Pope, but he could not get into the Milan College of Physicians. So what he did is he wrote this book on evidence-based medicine, the first one, a few years later, on the bad practice of medicine in common use. Just very shortly thereafter, he was admitted into the college, perhaps in fear that he'd write another book. He did become ultimately doctor to the Pope, and the Pope died. So there you go. Going forward a few hundred years, actually, this is really the first serious sort of intersection between quantitative medicine and we'll just say pathology, biologically based medicine. In 1835, a urologist looked at two methods to eliminate kidney stones. And he looked at the surgical approach, and he looked at 6,000. I mean, this is an unbelievable study. 1835, almost 6,000 patients, 78% survived surgery. The quote-unquote bloodless approach, conservative medical care, 260 patients. I don't know how they were selected. Almost 100% survival. Whoa. Now, these days, you think this would be pretty slam dunk, but then this was very, very controversial. So it was submitted to the National Academy of Sciences for review of these numbers. This was not a traditional way to look at the efficacy of medical therapies. In fact, it was basically the first time it had ever been done. So this is what that commission said. And to just give you some background, it was headed by a guy named Poisson, whose name is famous in statistics and mathematics as he was named after a distribution. That's a little joke. distribution is named after him. Anyways, one of the giants, and he headed it, but echoing issues of today, it has been told that on his commission was a surgeon who completely dominated the proceedings, and Poisson was quite cowed. Anyway, this is what the report of the commission said. It says, in statistical affairs, the first care above all else is to lose sight of the man taken in isolation in order to consider him only as a fraction of a species. It's necessary to strip him of his individuality in order to arrive at the elimination of all accidental effects. Those are, we call them confounders, that individuality would have been able to, individually actually, would have been able to introduce into the question. It's altogether in the domain, altogether different in the domain of medicine. It is not appropriate to elevate the human spirit to that mathematical certainty which is found only in astronomy. And to underscore this, he says, this is actually the surgeon on the panel, if 100 out of 1,000 boats perish, we still don't know which ships will be destroyed, and this is true. Mathematics cannot be used to forecast a determined event, but only establish a probability of a certain numerical proportion into two classes, but it's precisely this fact that makes it completely useless in medicine. The calculus of probabilities has been called good sense reduced to calculus, but one can perhaps ask whether good sense is calculable as the same as intelligence, the passions, the human affections, all that which affect human beings. So here you see this struggle between group-based knowledge and what you know about a particular patient sitting in front of you. And that was the fight that was going on here. Now this was echoed in the writings of a very famous French physiologist whose philosophy really became the foundation of medical education in the United States and is carried through today. And he was a champion. I mean, he was a reformer, and he wanted medical procedures to be based on sound scientific knowledge. But what he called sound scientific knowledge was a detailed understanding of the biology, and and physiology. There's certainly nothing wrong with that. But in doing so, he completely rejected statistics, anything based on a group. This, which echoes what you just saw, a great surgeon performs operation for stone. No coincidence that he said this, used this example. Later he makes a statistical summary and concludes that the mortality law for this operation is forward from what had preceded it, but not necessarily in the complete rejection of summaries of statistics. So what happened in the early part of the century with the formation of these schools, while also some of the scandals represented on my tie resulted in the formation of a council at the AMA, which eventually became the US FDA in 1938, and they were supposed to provide doctors with the means to discriminate between worthless therapies and valuable innovation. And the quote was, we need to counter influences that debauch our medical journals and taint our textbooks, which of course is the same struggle as today. So who were mistrusted in this world? Well, actually everybody. The pharmaceutical companies, the patients testifying that treatments work that maybe did or didn't, general medical practitioners, even research practitioners. So a whole bunch of methodologic things were introduced into medicine to try to get away from personal testimony of both the doctors and the patients that things worked. And these included these things: untreated control group, double blinding, randomization, objective outcomes, and objective methods of analysis and inference. And that's what we're going to talk more about right now. So we're going to talk about three things. Basically, what is probability? What does it mean? And then how is it used in statistical inference? Actually, that's mainly an evidence versus belief. So first of all, does anybody know where the term statistics comes from? Do you know what? Nothing. Don't worry. So it actually interesting. Statistics, in a sense, were actually discovered by the calculation recording of birth and death rates and other health outcomes in England. And it is literally numbers related to the state. So statistics. That's where it comes from. And it was only through this that even though we can't predict, who in this room is going to live or die in the next year or the next 5 years, what was amazing about these numbers when they were calculated in what were called the bills of mortality in England is that the numbers were unbelievably consistent from year to year, and that was marriages, divorces, deaths, births, weddings. Everything they recorded for the purpose of the state were remarkably stable. And when you think about it, this is mysterious, that out of what looks to be completely unpredictable phenomenon on an individual level is remarkably predictable and stable on a group level, in this case, the level of the state. And it was the recognition of that led to the development of a whole suite of methods for looking at these frequencies across the entire domain of human endeavor. But that's where the word comes from, and we're still struggling with its implications. So what is statistics? It's first of all a method to determine the best guess at the underlying truth, and it's also the method to determine the uncertainty in those guesses. So this echoes what I said about design. It also can be used to determine the weight of evidence for one possible truth versus another. So how do we measure our uncertainty? Well, of course, we do it with this thing called probability or risk, and we're all pretty comfortable with that. We talk about the chances of just about everything. But the fact is that this thing we call probability doesn't have a unique definition. And if I asked you to define it, you would really, really struggle to define it without using the word chance, risk, or probability. You would really be challenged. It's actually a very, very deep concept. And why is it deep? Let's think about it. If I asked you what your weight was, what your height was, what anything about you was, we could develop very quickly. You'd step on a scale, we'd take a ruler, we'd measure it in you, and that would be your. But if I asked, what's your probability? What's your risk of surviving the year? How would you figure that out? That's actually very tricky. What would you do? Well, in theory, you would go and you try to find a bunch of other people like you, right? And see what happened to them, maybe in the past, maybe recently. Who would you go to for that? Well, your insurance agent, right? They bet a lot of money on that. In fact, they're right. But do you want them to be your doctor? No. They might tell you the best way to increase the chances of living the next year to change your zip code, right? Well, that wouldn't work out too well. Actually, maybe it would, depending on where you live. I used to live in Baltimore, and probably I moved to California. I think my chance of surviving any given year probably increased. But in any case, that's not exactly medical advice. So it's really a very deep and interesting measure that runs counter to how we think about how we work or the world works, is that we measure it in other people and then we apply it to ourselves. And these other people are defined by what we call like. Well, what does like me mean? Same height? Same degrees? What does that mean? So you can immediately see that this is something very different than almost every other measure we make in biology and medicine. So here's some examples of all the different things we use probability for. Probability that Mrs. Jones will survive tomorrow's operation. Well, that we maybe can get a handle on. Probability the coin will fall 2 heads and 8 flips. Well, there's a formula for that. Probability that it will snow tomorrow. Probability that we'll go to war against North Korea. Probability that this talk will be stopped by an earthquake. Probability that the newest implant technology increases retention or causes bone loss. These are all things that we apply probability to, but each one we use a completely different methodology to figure out what that number is, and that number represents very, very different things. So why is it important that we look at the foundations of this thing that we call risk? Well, if probability is used to represent uncertainty, and statistics is the calculus of uncertainty, then one's approach to statistics is determined by the definition of probability that you use. And you might think, I don't have a definition of probability. I don't have ever thought about probability until you started talking up there. But you will find, actually, in some way, that you do, or you accept one implicitly. So there's actually 2 different kinds of probability. One is called epistemic. That is, it relates to the degree of knowledge you have about something. So you say there's a chance of something happen simply because your knowledge is incomplete about what might happen. And the other is called stochastic or frequencist or sort of objective, in that it's like the flipping of the coin. It's like a game. It's like a random mechanism. Now, epistemic probability has different forms, but fundamentally, it relates to how strong the evidence is for your belief that something will happen. And we use epistemic probability all the time. All the time. We use it when juries render verdicts. Guideline panels use it when they rate evidence as A, B, or C. That is epistemic. That's how much should you believe this evidence. So we use this language all the time. We use it when we make predictions or projections about whether Apple stock will go up or down. We use it all the time. So we think we don't have it, but we use it. But the question is, how should it be measured? What happens if people disagree? And is it scientific? These are real live questions, right? So what is this other thing? So it's a very uncomfortable thing to base the scientific enterprise on for exactly those reasons. So because of that, this other form of probability was developed. You'd think it was the first form, but it actually wasn't, which is called frequentist or frequency probability. And this is all based on sort of random processes like games, and it involves you imagining that whatever enterprise you're involved in is like a game. It's like spinning a roulette wheel. So doing a study, we say, imagine if you did this study many times, right? You've heard that lots of times. If we did this study many times, in fact, I said it before. But the fact is, that's just a thought experiment, because you're not going to do the study many times. You can spin a roulette wheel, but you can't do the study many times. So it's simply a figment of your imagination. So this has a problem, this form of frequency, because it's based on this thought experiment, and it feels real because you have formulas, but it depends critically on that thought experiment being real. So, and also the other critical thing about the frequency theory of probability, and you'll see how this comes into play in medicine, is that by definition, it doesn't apply to single events. So I can't talk about the probability it will snow tomorrow. I can't talk about the probability that this study result could have been larger. You're literally not allowed to do that, because the whole reason it was developed was to put probability on the same footing as height and weight and mass, that everybody looking at something would agree what the probability was. And that involved having many repetitions in front of them. So if we all looked at 5 out of 100, we all agree that we're all seeing 5 out of 100. But it was silent about what happened if you just had one flip of a coin, and we said, imagine if you flipped it 100 times, and imagine if you had 50 out of 100. So it was designed to be scientific, but there are many problems that actually make it literally scientific. So how does this fit into inference, which is what actually we're involved in when we look at studies? So what is statistical inference? Actually, I used to have 2 slides here. Let me just talk you through the medical inference is when you know what the condition is, and you can, if you know the underlying diagnosis, you can tell me what is likely to happen with a certain probability. The deductive direction is that direction. I tell you the diagnosis, you tell me what will happen. The inductive direction is the other way. The patient comes in with symptoms, and you try to figure out the diagnosis. That's actually much harder. And the same thing happens in statistics. If you tell me what's true, whether if there's a null hypothesis or there's a 5% effect or a 10% effect, I can tell you how often, using formulas, you will observe different degrees of effect. But the real question is the reverse direction, which is if you observe a 15% difference in cure rates, what is likely to be true? That is we'll call the inductive direction, and is very, very tricky. So what we see if we know the true proportion of something, we have formulas that tell us what we will observe and how often. But if we observe something, if we observe a batter who has a streak of 10 hits and 15 at bats, and you try to figure out, okay, what's their real batting average? That's the going backwards. That's statistical inference, and you can't use the same probability anymore, and that's the tricky part. Now, something called Bayes' theorem, which some of you may have heard, is a way of going backwards. That is, it says, given that the odds of something being true, and then you see data, this then changes those odds, and you have a new odds of that thing being true. So that's how Bayes' theorem was used, but the problem is, that the stating that the odds that something is true before you actually do the study is very tricky, and not everybody will agree on it. It's that thing, that degree of belief probability. So it's not clear that that's scientific in a certain way, in that everybody would agree. So there are a variety of scientists over the years that tried to develop a way of mathematical reasoning that didn't use that didn't require you to say, how likely do you think this is going to be true? And now you see it, have an experiment, and then you change your probabilities. So this is the conceptual framework for analyzing research that's suggested by Bayes' theorem, that you have evidence, that is what the data say, and then you have belief based on that evidence, that depends on the evidence from that study plus everything you know about the phenomenon, and then you have an action that you take based on your belief and the consequences of your action. And that this is what studies do. They bring you from a prior state of knowledge about how certain a therapy is that works to a final, sounds like Aflac, to a new state of knowledge after the study, and that's what we call strength of evidence. So this is the way things are broken down using Bayes' theorem, but this is not the language of traditional statistics. So the language of traditional statistics is p-values and hypothesis tests and confidence intervals. Okay, it doesn't talk about belief, certainly doesn't talk about action. And this is where some of the confusion that we have today comes from. So let's talk about these things called p-values. This is this guy, R.A. Fisher, who actually very few people know about, even though I told you about him before, one of the real giants of the 20th century. And he developed the p-value. And what do you think, anybody here know what it means? Does it mean probability, plausibility, possibility? Probably think that's what it means. Actually, it did come out of probability. It was the probability value. So what is it? What is its definition? So I'm just going to say what its definition is, and many of you know it, but this doesn't immediately tell you really what it means or how it should be used. So here's the distribution of all possible outcomes that we could see under the null hypothesis. So to dress this up a little so it makes sense, null hypothesis could be, a treatment doesn't work. That is, the outcome is the same in this group as that other group, and yet you can see a whole range of treatment differences even if the null hypothesis was true, just by chance. So what you want to know is it so unlikely under the null hypothesis that the null hypothesis is not a tenable explanation for what you're seeing? That is, it couldn't happen by chance. So you observe something out here, which might be a 20% treatment difference, and what we do is we take the area under that curve, we say the probability of what you saw plus more extreme results under the null hypothesis, and that's the p-value. So there it is, the probability of getting a result as or more extreme than what you saw, than the observed result, if the null hypothesis of only chance operating is true. Okay? So think about it. It says if the hypothesis that the treatment is useless is true, this is the probability that I'd see something this large or this treatment effect or something more extreme. So since the p-value is calculated assuming the null hypothesis is true, it can't represent the probability that the null hypothesis, that it can't represent simultaneously the probability of the null hypothesis being true. It's assumed to calculate the number that there is no effect. And then this calculates the probability of the data that you saw, or more extreme data. But what are we actually interested in science? We're not just interested in the probability of our data. We're interested in this thing, the probability that there's no effect. That's what we're interested in, the probability that our treatments do or do not work. So this is the reverse. It says, if the treatment doesn't work, then we will see something this extreme or more. It does not say, given what we saw, what's the probability that the treatment works? It doesn't tell you the reverse. That's in the realm of Bayes' theorem, but Bayes' theorem is based on the theory of probability that early scientists, Fisher included, tried to get away from because it seemed unscientific. So I've published, and many other folks have published, lots and lots of lists of things that the p-value is not, and it's almost nothing sensible that you would think. It's not the probability of the null hypothesis, or that the treatment doesn't work. It's not the probability that you'll make a type 1 error if you reject the null hypothesis. It's not the probability that chance produced the observed result. It's almost nothing that you can think of. And that's the problem. So what we really want in science is, what's the probability of the truth? And the p-value doesn't give us that. I don't want to go through all these things. So it was also a guideline for inference and not for behavior. And I want to give you a quote from Fisher when he described what the 0.05 level meant. Now, what was very clear about Fisher was that he just wanted this to be a tool. He just wanted to be one of a broad suite of tools to be used. He didn't want it to be used rigidly. And this is what he said. If 1 in 20 does not seem high enough odds, we may perhaps prefer 1 in 50 or 1 in 100. Personally, the writer prefers to set a low standard of significance. So he saw this as a low level of evidence, and to ignore entirely all results which fail to reach this level. But did he think it represented truth? No. And this next sentence is maybe the most important. A scientific fact should be regarded as experimentally established only if a properly designed experiment rarely fails to give this level of significance. So what does that mean? It means that if you do the experiment over and over and over, and it's significant at this level every time, then you believe it. But he purposely designed it to have a very low threshold, because he didn't want to miss things that worked. So if he made it too stringent, you could miss treatments that worked. So he said, okay, let's set a low bar, and then if it passes that bar, we'll do the experiment again. And if it passes that, we'll do it again. And if it keeps coming up, then we believe it. So it was